Thanks to everyone for standing by, and welcome to the Radiopharm Theranostics Investor Webinar and Q&A discussing the brain tumour technology recently added to Radiopharm's portfolio. All participants are in a listen-only mode. There will be a presentation followed by some Q&A based on questions that have been submitted via email and live during the session. If you'd like to submit a question, please do so using the Q&A function within the Zoom app. On the webinar today, we have from Radiopharm Theranostics, we have Managing Director and CEO Riccardo Canevari, and joining him is the inventor of the technology, Dr. Susann Brady-Kalnay. Dr. Brady-Kalnay is a professor and distinguished faculty researcher in the Department of Molecular Biology and Microbiology at Case Western University, as well as being a professor of neurosciences, pathology, and member of the Case Comprehensive Cancer Centre. She is also the founder and CSO of diagnostic and prognostic technology business NeoIndicate. The presentation today will last for approximately 15 minutes, with approximately 10 minutes for the Q&A thereafter. To begin, I'll hand it over to Riccardo. Please go ahead.
Thanks a lot, Matt, and thanks for everybody that is listening and following this webinar. Well, we are excited. We are excited by our latest add to our portfolio. We believe it's an incredible technology in an area of very high unmet medical need, and really this combination of a great technology in an area where it's important to find new possibility of imaging and therapies for patient is really important for us. So I already said it's a promising imaging and therapeutic radiopharmaceutical, has been studied deeply in preclinical in a leading US university, Case Western in Ohio. It is specific, it's kind of a unique technology that is targeting the so-called PTPµ-X, so it's a fragment of a receptor that is expressed in the cancer cell. And what makes this very interesting, that this receptor is not expressed in the healthy tissue. So the science behind is telling us that this product has the potential to be very and extremely selective to the cancer cell, but leaving almost untouched the healthy tissue. And of course, this is great to balance efficacy and potential safety. A lot has been done in the preclinical model in different tumor types, with a key focus in a brain cancer that is called glioblastoma, where we see the potential to be our leading indication. The amount of information we have and study that has been performed allow us to predict that we can be in phase 1 probably in 12 months from now, so very fast. In order to do that, we need to manufacture the peptide, and we plan to do in the next few months before the end of the year. And of course, because we are a radiopharmaceutical company, we need an isotope, in this case lutetium-177. And probably some of you might have seen a very recent announcement from Radiopharm Theranostics, and we complete an agreement with a company from Israel, and we have now access to lutetium-177. So we already have the technology, we already have the isotope, so we are in a very good shape to complete and to start our program. We believe that considering the high potential of the technology, we had a very attractive commercial terms, and we will continue to work with Case Western and with Professor Susann Brady with a research agreement, because we really like the experience and the expertise to continue in this area, so that we can develop this molecule in the best way. And of course, always when you license a molecule, you do a proper due diligence in terms of the patent. The patent is solid, is 2037 and beyond, so we have many years in front of us. But I'm really excited to have here Professor Susann Brady, so I will hand over to her that she can tell us much more about this exciting technology.
Yeah, so hello. I have discovered a new molecular imaging agent that we can use on cancer cells, and it's based upon something that happens in the tumor microenvironment where a specific form of PTPµ is generated. And we developed agents that can bind to that tumor-specific form of PTPµ. Next slide. So we were able to use this tumor-specific agent, and you can see that it binds to tumor cells, but it doesn't bind to normal brain. And it's as good as if you labeled the tumor cells specifically with the green fluorescent protein, as you can see with the red staining and the overlay image. Next slide. So we were able to add this imaging agent to an animal model, and what you can see is within minutes, it's bound to the tumor. It's recognized it. It travels through the body and specifically binds to the tumor, and it's stable for a number of hours. Next slide. So we've also used this when tumors are in the brain, and you can see that the green is the main tumor mass, and the yellow are all the invading migrating cells that surgeons can't see right now. So the purple shows you of the yellow cells, which ones are labeled by PTPµ agents, and you can see that all of the tumor cells are labeled in pink, regardless of how far they are from that main tumor site. And it can be millimeters away in these mouse tumors. So it recognizes the main tumor mass and all the invading migrating cells that have moved away from that mass. Next slide. So
Thanks a
we
lot.
have a
Thanks a lot, Professor Brady. I think it's exciting, and we discussed together, of course, also what can we do beyond preclinical. Now we want to do a phase 1 clinical trial. We want to go to patient very very fast, and for this reason we select and we thought that glioblastoma is the right type of brain cancer where we should start the trial. The reason is unmet medical need. Unfortunately, for those patients, there are limited options, and prognosis is very poor. We also believe that because glioblastoma is considered an orphan indication, the approach from the FDA and from the regulatory agency will be very positive, and we have opportunity to have an accelerated plan, really to do the trial as fast as possible to potentially bring this molecule and the technology to a broader number of patients going through the phase of the development. At the moment, there are no radiopharmaceutical therapy approved in glioblastoma. There are some in development, and we believe that with this technology and with our approach, we really have the potential to be the first radiopharmaceutical therapy for patients with glioblastoma, and we really are excited by this possibility. And for those of you that are less familiar with the disease, is really a devastating disease. Usually start with symptoms, and then the patient go and do a scan to understand what is going on in him or in her. And unfortunately, when you detect that there is glioblastoma, is a very, I would say, devastating news for the patient. The first approach is to go for surgery. You try to remove the tumor, but of course, as you can imagine, surgery in the brain is very difficult and very delicate, and is not always possible to remove all the cancer cells that are near the main tumor. So what you usually do after surgery, you use external beam radiation and chemotherapy to try to limit the growth of the cancer cell that you were not able to take out with surgery. So all of this description lead, of course, to a poor prognosis. Is a very highly invasive cancer type. Only 7 patients out of 100 are still alive after 5 years from the diagnosis, and there are limited therapeutic solutions. That's why we really felt the responsibility, and if you want also the excitement to do something new in this space, and combining this great technology with therapeutic radioisotope might be a winning approach for this disease. So in summary, and we of course we like to answer your question that I assume that will be mainly for Professor Brady. But in summary, this technology has imaging and therapeutic potential, has been licensed from a major and leading institution. The mechanism of action was very well explained. It's targeting the PTPµ expression that is in the cancer cell, but not in the healthy tissue. We believe that we can have accelerated development plan with this technology and the potential to be the first in class. It has a strong patent and IP. At the moment, there is limited competition, and we believe in the potential of this molecule. And just to put in the context of what Radiopharm Theranostics is doing, this is our latest in-licensing, is our sixth pillar, adding to what is already present in our very deep pipeline. So we are very pleased that we have the PTPµ, this peptide that we plan to do imaging with gallium and then therapy with lutetium in this very important indication. And with this, I will hand over to Matt and open to any question might be from the platform. Thank you for following the webinar.
Thank you very much, Riccardo, and thanks to Susan as well. We'll move on to the Q&A session. The questions are based on those sent through via email, and we'll take some live questions as well. So if you do have a question, please feel free to type it in using the Q&A function within Zoom. Riccardo, I'll start off with one for you, which was just to ask, you've obviously got quite a few different programs now. How are you prioritising the different assets in the portfolio for RAD?
Yeah, thank you for the question. It's a very important one. What is interesting that, of course, each of these technology that we have in our portfolio is in a different stage. So we have some technology that are in phase 2, some other are in phase 1, and the PTPµ brain cancer technology is in preclinical. So it's really about doing different things for every different project. We don't have to compromise. We can really develop our 6 platform in parallel, because all are important and all require different steps. So we are very committed to bring our pipeline to clinical trial as soon as possible, and the ones that are in clinical trial to continue with the operation.
Thank you, Riccardo. I've got a question for Susan. What are the challenges in linking the isotope to the peptide?
There are various strategies to do it, and we have a successful strategy that we've used in the past, which includes using a chelating molecule to actually add the chelator to the imaging agent, and then you can complex in, it's called a radioisotope or any metal.
Great, and I think this would be considered a follow-up to that, which is, so if the peptide crosses the blood-brain barrier, is it correct that this will be delivered as a systemic therapy?
Yes, that is correct. We use it systemically. We've done that successfully with a number of imaging modalities, including fluorescence and MRI, and now we would like to repeat that with PET imaging and radioisotopes.
Great. Another more general question for you, Susan. Can you talk a bit more about how long you've been working on the technology and why you think it's particularly exciting given your experience and what you've seen in the field?
Yes, so actually, I started working on this class of proteins over 30 years ago as a graduate student and have continued in this field. But what's become very exciting in recent years is that I'm able to use these peptides as imaging agents and to deliver therapeutics to tumors. So we're very excited about this possibility because the direct delivery of a radioisotope specifically to tumor cells that are invasive has not been done in the field, and that's a very exciting possibility to us.
Great, and one more for you, Susan, at least. Can you elaborate on why you are happy to partner or keen to partner with Radiopharm to further the work you've done on the technology?
So they are world leaders in this type of approach. So there are many experts on their team that deliver radiotherapeutics and radioisotopes to locations. And so us being able to partner with RAD is just phenomenal. We're very excited about the possibility because we get to work with world experts in this space.
Great. And another question that's just come through, is the IP around the composition of matter for the peptide?
Actually, our IP is very, very broad. So it's actually to the platform of technologies, to an entire set of proteins called the immunoglobulin superfamily. And it has both the agents, composition of matter, and methods in the patent. So they're very far reaching, and we're extremely lucky to be able to move this platform forward with RAD.
Thanks, Susan. Riccardo, this might be more of a question for you. Could you add your targeted timeframe for the clinical development plan and your manufacturing arrangements in San Diego, please?
Yes, with pleasure. So in terms of manufacturing, we already, of course, if there is preclinical ongoing, is because the peptide is available. But we want to be sure that we produce this peptide with the right quality that are requests when you move from mouse model to human. So we are selecting the right producer, the right CMO, to be sure that we have the right quantity and the right quality of this peptide. And we believe, because we are in advanced discussion, that we can close this agreement by December. Regarding the clinical development plan, there is still some preclinical work to be finished, and Professor Brady will continue her high quality work in this direction, and we plan to start engaging with FDA around September or October of this year, so that we can have a phase 1 clinical trial in about 12 months from now. So very fast, considering we just in-licensed this asset.
Thanks, Riccardo. And just one final one. It'd be good just to get your thoughts on why you brought the technology into Radiopharm and why you're excited about it.
Well, in general, I'm very excited about the approach. Now, if you think there has been very important progresses in oncology in the last 20, 30 years, and beyond the traditional approach that is chemotherapy and targeted therapy, more recently we saw emerging area like immuno-oncology or cell and gene therapy. Well, all these are important, but we believe and we ask ourselves, okay, what is the next big innovation in oncology? And we believe that these are the radiopharmaceutical therapy. So the possibility to deliver in an extremely selective way radiation or high energy to the cancer cell, but not to the healthy tissue, has been proven to be successful already in a couple of indications like neuroendocrine tumor and prostate cancer. So the approach of Radiopharm Theranostics is really to find the best molecule outside, like this peptide targeting PTPµ, extremely selective, link with the right isotope and go for clinical development. I believe that this can be absolutely a successful approach for patients living with oncological disease. And this is what is exciting us, the potential to give our contribution in improving outcomes.
Thanks for that, Riccardo. We'll wrap it up there. I just want to say thanks again to Susan for joining us in the middle of, I know she's got a very busy week, so that's much appreciated for taking the time out. And thanks to everyone for joining the session today. A replay will be available shortly after the conclusion at the same link. So thanks again, and thanks Riccardo and Susan.
Thank you, everybody.